Okay, thank you very much. Good morning, everybody. My name is Girma Nigussie. I am a PhD student from the University of Gondar, Ethiopia, and I am here by the opportunity given by the Erasmus and UVA. So, in this occasion, I want to thank to the Erasmus and UVA, and and also I am really happy to present my work on this respected conference. So my work is entitled with knowledge of wild edible plants across social groups in northern Ethiopia. So as the introduction, as you know, wild edible plants are very important as for economically, ecologically, and as in terms of food security. However, these wild fruits are important, but in the study area, the diversity of the species and the knowledge of perception on this wild fruit is not studied in the selected district area. So the aim is to document wild fruit diversity and analyze how ethnobotanical knowledge varies among social groups. So the study was conducted in the Abergele district, which is northern part of Ethiopia, and the area is dry land or drought-prone area. So how we conduct is we collect the data from 387 key informants with semi-structured interviews, focus group discussion, and guided field work to analyze the habitats and the species types of species. And the analysis was done by documentation and statistical. We used the statistical instruments like ANOVA, correlation, multivariate, Paranova, and NMADs are to explore the knowledge patterns. So we have the 5 key findings. The first finding was the area is rich with different types of plant species. We document 40 wild edible plants within 22 families, and the growth forms was almost the majority was tree and shrubs, and primary use of the wild edible plants were 70% of the species was considered as fresh or raw, and the habitat or the distribution of these wild fruits is we can found in natural forest, grazing land, river banks, and farm edges. So as example, these species are this first, and this is Diasporus, and this is the CGM, and this is the Zymania. The key finding too was knowledge is not evenly distributed among the studied area. As you can see the figure, the key informant has almost 2 ways of the general informant, and also gender, occupation, family size, and age is a factor of the knowledge because male has high knowledge than the female. This is because of the interaction with the rural areas. The male has more interaction with the wild fruits. So because of that, the male has no more than the female. So with status, age, family size also affects the knowledge of the wild plants. The other key finding was multivariate analysis of knowledge pattern. As you can see the figure, there is clear difference cluster of between key and general informants. The general informant was scattered, very scattered, which means they know the less number of wild edible plants, and also general informant one knows the knowledge of general informant one is different from the other general informant, not in the number but also the type of species. But here the key informant is almost the same, and they know more knowledge of they have more knowledge of the wild edible plants, and they also the same the knowledge of the wild edible plants. They know more and they know the same types of species. And the other finding was seasonal availability. This wild fruit was more, we can find the matured wild fruit from June to October. So the red line is the occurrence of wild edible plants. The blue color was represents the preference of the local community to collect, because these 3 months are critical for the local community. So the other findings was the threats. These are the major threats of these wild protests: deforestation, agricultural expansion, settlement expansion, overgrazing, etc. are the major threats of the local. So when we come to the conclusion, summary of the key insights were: wild edible plants are indispensable for food security and cultural resilience. Ethnobotanical knowledge is rich but unevenly distributed. Wild edible plants serve as seasonal safety nets and have livelihoods. These species face several anthropogenic factors. So I want to acknowledge these individuals because this individual has a great role in my PhD journey. Professor Pablo, Dr. Gonfa, Dr. Asmamo, Dr. Daniel, Dr. Hermes, and also my mentor, Marina, and my colleague, Dr. Joe, Dr. Eric was, their role was very critical for my PhD. So I would like to thank you for your attention also. Thank you.